After 12 years of searching, Monsanto found a soil bacteria that is naturally immune to Roundup herbicide. Their goal was to genetically engineer DNA from these bacteria into various plants. They cut out a sequence of DNA that is resistant to Roundup. But if this DNA sequence alone is inserted into a corn plant, it will have no effect. So the next step involves E. coli bacteria. Gaps are created in the E. coli DNA, and when the two test tubes are mixed together, some of the E. coli DNA recombines with the Roundup-resistant bacteria. Then the technicians smuggle the engineered DNA into the cells of the corn plant they want to modify. Cells will naturally reject foreign DNA, so they developed a method using soil bacteria that causes tumors in plants. They use this bacteria to ferry the engineered DNA into the plant's nucleus. There are also two other methods used to get the engineered DNA through the cell wall. One uses a stream of electricity to create tiny holes in the plant cells so they become vulnerable to infiltration by foreign DNA. Another is the gene gun, which blasts particles of gold coated with engineered DNA into the plant cells. Each of these three methods needs a promoter gene that turns on the desired characteristics. The promoter gene is often extracted from the cauliflower mosaic virus.
This capacity of bacteria and viruses to invade mammals in different ways is what really has a lot of people edgy about biotechnology, because that's really what the tools are all about. In order to move genetic material from one organism to another that don't normally cross, you've got to sort of behave like bacteria and viruses and invade into the cells and become established, just like a virus must become established.
And they do one more thing. They attach to that an antibiotic marker system.
The antibiotic marker is a gene which is naturally resistant to a specific antibiotic, so later they can test whether the genetic cassette is being expressed.
The most cataclysmic force in the food system right now is the fact that the medical community is just terrified about the loss of antibiotics. No one really understands how using antibiotic marker genes in genetic engineering techniques might contribute to the problem. The biomedical community on a worldwide basis is absolutely focused on this problem now.
As we move on into this so-called biotech revolution and we start producing more and more transgenic manipulations, we'll start seeing pieces of DNA interacting with each other in ways that are totally unpredictable.
We have now learned that genes function in complex networks, and a single gene can express or influence many traits.
I think this is probably the largest biological experiment humanity has ever